we're going to explain what cloning is. Do you know that we can make an animal but two different breeds? If you don't know the answer is yes, by cloning. Do you know what cloning is?
A clone is any cell or individual which is identical to another. In biology, cloning is the process of producing one or more genetically identical individuals. With whole individuals, it usually means deliberate production of identical copies. This was first achieved in mammals with the famous Dolly the sheep. Many researchers tried to clone different types of animals. The first researcher who tried cloning is Hans Driesch. The first instance of cloning dated back over 100 years ago in 1885 with cloning of a sea urchin. Second is Hans Spemann, who conducted the first nuclear transfer experiment in 1901 by splitting a two-celled salamander embryo into separate cells using a single strand of hair from his own child's head. In 1952, Robert Briggs and Thomas King used the nuclear transfer technology found by Spemann to clone frogs from adult donor cells. But the most famous cloned animal was cloned by Ian Wilmot. Through many years of researching cloning, Ian Wilmot cloned the first mammal from an adult cell in 1997 with a sheep named Dolly. The cloning of Dolly was breakthrough in cloning research and started many public debates about cloning of humans. So what is the process of cloning? The cloning process usually involves removing genetic information, known as DNA, from a cell taken from one animal and placing this into an unfertilized egg that has had its own DNA removed. This egg is artificially stimulated to start developing into embryo and placed into surrogate mother. The importance of cloning. Cloning stem cells to make identical ones as individual could be used for medical reasons, for possibly growing whole new organs without the worry of the stem cells being seen as foreign in the body and immune system being triggered. Cloning stem cells from an individual with a disease. Let scientists and researchers understand the disease and develop a treatment for it. In use by police. Police have also used cloning in investigation for identification. This is called genetic fingerprinting. This process is done by extracting DNA from body fluid such as blood or saliva and cutting the DNA with restriction enzymes.